Hi, my name is Fatima Shireen, second year microbiology, Central University of Istanbul. Today we're gonna discuss about bioremediation of toxic waste sites. Before talking about bioremediation, we will talk about toxic waste and sites. The urbanization and industrialization have brought numerous pollutants into the world, especially in the agricultural field. We are using fertilizers, pesticides, herbicides. They are causing pollutions like aluminium, copper, zinc, nickel, lead, etc. Similarly, untreated pollutants from wastewater of agri-food industries disposed into river, canals, and other water bodies are harmful effect on environment that to these sites, I wanted to say that there is a lot of toxic waste sites, including crude oil industry, mining industry, and also there is radioactive materials, which is also a toxic waste. In this presentation, I'm dealing with three toxic waste sites and the bioremediation technique that used to treat this toxic waste site. And first here, dealing with acid mine drainage. So you can see the pictures of acid mine drainage from a gold mine dump. This is exposed to the inhabitants and it have a high risk of sulfates, toxic metals, and radiations. These pictures was from a report of Environmental News South Africa from 2016. Acid mine drainage is an extremely environmental hazardous byproduct of mine, often from natural leaching of groundwater, excess precipitation, or flooding of mine. These water interact with minerals and rocks containing sulfur, commonly pyrites, and it creates sulfuric acid and dissolved ions through a series of oxidation reactions. This increasing pH, like through mixing with more natural water, can cause precipitation of metal oxides. When the liquid escapes the mine area, perhaps through leaching, unsealed bottles, overflow, or containment failure. This acidic addition alone create unsustainable condition for wildlife and plants. In this case, we are talking about the released acid mine drainage is having sulfur and it react with pyrates, it will form sulfuric acid and it can oxidize ions from iron and it can precipitate metal oxides from the water. When we add cow manure or that kind of organic material with the sulfate reducing bacteria to the mine, it will act on the sulfate and it will form sulfate and also it will reduce the sulfuric acid to form rehydronium ions so that we can form a pH buffer bicarbonate. So it will maintain the pH of the acid mine drainage and also neutralize the pH. This is an in situ bioremediation bioreactor using sulfur reducing bacteria. So in this model, we are using substrate as 7 is to 2 is to 1 ratio. 7 is cow manure and 2 is decomposed wood chips and 1 is alfalfa straw. The substrate will be installed in this shaft along with the sulfur reducing bacteria. In this model, the substrate is added in three places. One is in the main shaft, which is here, and another one is the downwards, and it will come first. It will move through the first plug of substrate that we have plugged here, and after that, it will go to the second and third plug, and it will move further away, and it will move to the drainage. From here, these tunnel chambers and portal chambers, we can identify that how much it is cleared, how much toxicity is removed from this drainage. There is several methods and models of acid mine drainage remediation system, and it's one of the model that I'm explained here. The second toxic waste that we are dealing is polycyclic aromatic hydrocarbons. Oil spillage is a serious threat to all part of the ecosystem during extraction, transportation, storage, and distribution operations. Crude oil and its refined products are frequently spilled, causing oil pollution. Crude oil is a mixture of various organic and inorganic compounds that include alkanes, cycloalkanes, aromatics, polyaromatics, and additional chemicals like nitrogen, sulfur, small quantities of metal like nickel, vanadium, and sometimes water. The major methods of bioremediation of crude oil contaminated soil is land farming, composting, biostimulation, bioaugmentation, and phytoremediation. For the land farming, we need nutrient addition and soil mixing. It means when we take a large amount of contaminated soil and we add a layer of uh, sludge in it and we mix the soil and we will stimulate the microbial population and it start degrading. In composting, we all know we will add nutrients such as manure or dried leaves, etc. And we will provide the moisture and temperature. And the annual manure amendment will be formed and the vermicomposting will be started and all the high amount of carbons and alkenes will be degraded. And in biostimulation, we need nutrient addition like nitrogen, phosphorus, and we need a terminal electron acceptor like O2 or terminal electron donor and a fatty acid. In this case, mushroom cultivation substrate, and these will start degrading the carbon in the crude oil contaminated soil. And other one is bioaugmentation. Bioaugmentation is nothing but adding genetically modified bacteria into the microbe population. It will enhance the performance of the microbia. And other one is phytoremediation. Phytoremediation is using plants. We will reduce the contamination. So the plant's root can absorb um, contaminants. It will reduce the carbon. These are the bioremediation technologies used for hydrocarbon contamination. We already discussed it. We are now comparing technology, key point, advantage, and disadvantage. For natural attenuation, it using indigenous microorganism and natural condition. And advantages, it's a cheapest technology, but the disadvantage is it will take a long-term monitoring, but and also it's not always successful. And another one is bioaugmentation. It is also addition of hydrocarbon degrading microorganism, as we said, genetically modified microorganism. Using high biomass of polycyclic aromatic hydrocarbon degradation microorganism. And the disadvantage is it can it is a it have poor adaptation. The bacteria will have poor adaptation to the site and also it changes the natural microbial structure. And biostimulation, the key point is addition of nutrient in it. We all know when we add bacteria to it, when we have to stimulate the bacteria, we will add additional substance for the bacteria. That is biostimulation. Uh, so the advantage is more efficient than natural attenuation, but not always successful. In the case of phytoremediation, it is using the plant and their associated microorganism in it. It supports, in the case of phytoremediation, using plants and their associated microorganism, the advantages supports hydrocarbon native microorganism within the plant root. The disadvantage is when this uptaking the toxic substance, the plant is getting toxic. Maybe the plant can get the toxic effect and eventually it may be end up in a bad situation. Bioventing is another institute biodegradation technique that relies on maintaining and enhancing the activity of indigenous soil microbes by providing oxygen to improve aerobic degradation of xenobiotics. We can see here there is a saturated and unsaturated zone. In the unsaturated zone, we can see the contaminated zone. So the contaminated zone also have the bacteria which can degrade the pollutant. So what we will do is we will inject oxygen and we will withdraw the oxygen from both sides. So when we inject oxygen from one side, the oxygen will pass through the contaminant and we will withdraw the oxygen from the other side. So the bacteria can get enough oxygen and it can act correctly and it can remove or degrade the pollutants. These are some of the fungal genera and polycyclic aromatic hydrocarbons which they act, and these are the substrate or environment which they will find. And one of them is Aspergillus niger. We can see it is acted on anthracene in a waste cooking oil. And also Aspergillus oryzae and Mucor irregularis are found out to be acting on complex hydrocarbons in Nigerian crude oil polluted sites. And Aspergillus tetrus are found out to be acting on phenethane in soil. And like the previous slide, these are some bacterial genera and the hydrocarbons which they were acting and the environment which they were found. Here we can see Comomonas testosterone were acting on several polycyclic aromatic hydrocarbons in polluted areas, like that Rhodococcus species are found out to be very excellent for this degradation of hydrocarbons in marine environment, etc. In this picture, we can see induce phytotoxicity and inhibit seed germination. For this test, we are taking few petroleum contaminated soil and first we are started to germinate a seed, but the seed is not germinating. You can see there just because the soil is too much polluted with the hydrocarbons. And in the second test, they are doing same contaminated soil but adding a Pseudomonas strain in it, SA3, and we can see the seed started to germinate. And the other test, the last one, they added the same petroleum contaminated soil and strain Pseudomonas SA3 and biosulfactant to it. So we can clearly see that the seed is completely germinating. So this means the Pseudomonas species and biosulfactant act together. It can degrade the crude oil contamination. Third toxic waste is radioactive waste. Radioactive waste is a discharge from many sources, including nuclear weapons, uranium mining, power plant effluent, and medical research laboratories. Radioactive waste is filled with persistent unstable elements or radionuclides which emit ionizing radiations as they decay. Radioactive elements have no stable isotopes, high molecular weight, and fall into groups such as actinides. They are unstable because their atoms spontaneously discharge, emit and both particles and energy as they transform into more stable form. Bioremediation of radioactive waste is mainly done by radiation-resistant bacteria. This is genetically engineered to metabolize radioactive waste. Deinococcus radiodurannus can resist extremely high concentration of radioactive due to potent antioxidant and enhanced DNA repair mechanism. These bacteria can resist extreme concentration of radioactivity just because it have an extreme enhanced repair mechanism of DNA and potent antioxidants. We can modify this bacteria to create a radioactive metabolizing bacteria. These are the picture of this is the electron microscopic picture of the Deinococcus radiodurannus. These are some of the mechanisms which the bacteria use to deactivate or reduce the effect of radioactive metals. In bioreduction, microorganisms inactivate some radionuclides by adding electrons to them and converting the radionuclides from soluble to insoluble state. This is two types, direct and indirect. Bioaccumulation is nothing but building up of these kind of elements inside the bacteria, but some of them are really effective for the bacterial growth. Actually, these bacteria are taking the all radioactive elements and putting it into their cells, so it is conserving these things. Some of them are really helpful for them. The bacterial bioabsorption, we can see the bacterial surface is completely covered with a lot of negative charged substance like sulfate, sulfides, and phosphates. This can attract these chemicals to them, and it will attach to them, and it will remove the substance from this environment. Bioabsorption is a reversible procedure when the substance or elements which is in the surface of the bacteria can attach with this uranium or another radioactive metal. At the same time, they can detach with them too. Biomineralization is an insoluble catalyzation of metals. Metal precipitate with ligands such as sulfides, carbonates, phosphates, and hydroxides that are enzymatic catalysts, which is present in the microbial cell wall. This is a summary of strategies used by various species and genera of microorganisms to bioremediate radionuclides. From this table, we can say that geobacter can use for the bioreduction of plutonium, like Bacillus mycoides can use for the bioreduction of plutonium. Pseudomonas species can do bioabsorption and bioaccumulation of uranium. At the same time, Bacillus subtilis can do the bioaccumulation of thorium. This is an overall table of bioreduction, biomineralization, bioabsorption, and bioaccumulation, and the bacteria which can do these procedures to eliminate the elements like plutonium, neptunium, thorium, etc. And these are my references. So that's it. So I'm concluding my presentation. Thank you. Thank you for all.